Hello students, this is Fazan Mirza. We are discussing the process of cell signaling and how the cells communicate through the cell membrane and the cell signaling molecules. The cells communicate with one another using cell signaling molecules which travel in your blood or in your body fluids, and they reach from the region where they got released to the region where they need to act, and they bind to the receptor present on the membrane, and they bring about a certain change in that cell. The cell signaling involves basic components that you have studied in your O levels as well or in your previous classes. That your receptor, basically accepts a stimulus or it monitors a stimulus or a signal. The message is transmitted, the signal is transmitted to the target or the effector, and the response comes about. The receptors are present on the cell membrane. They receive the signals and the stimulus. The cell releases a cell signaling molecule, which might be a hormone or a neurotransmitter. But in this case, which we are discussing right now, the hormone or the neurotransmitter will be water soluble. It will be polar. It can be non-polar as well, but the non-polar will be functioning through a separate pathway for which we can discuss later. But right now, we'll be restricting ourselves to polar cell signaling molecules. They work by the process of the second messenger. They activate a second messenger. The polar cell signaling molecule itself is called as the first messenger. It will travel throughout the body to or throughout the region from where it's secreted to find its target or the effector. The cell signaling molecule attached to the receptor on the cell membrane of that target or the effector. The cell might be like very close to it, or the cell might be in a distant location to another in another organ. The target or the effector then responds by either secretion or movement or metabolic change or activating certain gene. This is how the response occurs because of cell signaling molecule. In a cell signaling involving second messenger, this basically is about a polar molecule, which is a cell signaling molecule. When it attaches to the protein receptor present on the target tissue, the signaling molecule attachment to the protein receptor on the target tissue triggers the G protein, which is attached to the underside of the same protein receptors in the cytoplasmic surface. The G protein shuttles along the phospholipid bilayer and attaches itself to an enzyme that makes the second messenger. This enzyme will also be located within the phospholipid bilayer of the target cell or the target organ. The enzyme that makes the second messenger becomes activated. It activates second messengers, which are small soluble signaling molecules that spread throughout the cell, greatly amplifying the signal. So several second messenger molecules will be made here. These second messenger molecules will go on and then they will activate enzymes that initially were inactive. For example, initially we saw that the enzyme that made the second messenger was inactive, but the attachment of the G protein triggered its activation. So the release of the second messenger activates the enzymes which initially were inactive in the cytoplasm. These activated enzyme will go on and activate even more set of enzymes which initially were still inactive. So once those enzymes have been activated in multiple tiers, these enzymes will be activated one after the other, and eventually the response will come about. The cell will start to either secrete a substance which the cell signaling molecule wanted it to, either the cell will start to undergo transcription. Transcription is just it switches on certain genes which initially were silent, so it will start producing a certain protein, or the cell will start to perform some kind of movement of vesicles within the cell, or the cell will start to move by itself, for example muscle contraction, or probably the metabolic change can occur. Metabolic change involves a change in the metabolic reactions. This can be, for example, in the presence of the hormone glucagon. The liver starts to release the stored glycogen by converting it back into glucose. So when the hormone glucagon attaches to the protein receptor of the liver cells, a G protein activates the second messenger molecule, second messenger enzyme that activates several second messengers, and those second messengers they will go on activate enzymes, and the activated enzyme will activate even more enzymes, and then even more enzyme will be activated, and eventually an enzyme will be activated that can break down glycogen into glucose. So the glucose which is formed here will start to move out from the liver cells and into the bloodstream, and this is how glucagon increases your blood glucose level back to normal in cases of fasting. So this is about second messenger molecules. If the second messenger molecule was a lipid soluble molecule and not a polar molecule, it wouldn't need a second messenger. It would just penetrate through the phospholipid bilayer. It would go and trigger the genes in the nucleus right away because it can travel through the phospholipid bilayer. It is just the polar cell signaling molecules that act as first messengers, and they cannot enter the phospholipid bilayer, so they have to work by the second messenger. That's it. Thank you so much.